Okay, so this is a continuation of part one of this. I'm just going to make this a shorter video and try to finish up part one. Okay, we've got crossing over occurring here and the chiasma formation. Remember, that's that structure that actually forms during crossing over, during the exchange of genetic information of the homologous chromosomes during prophase one. It results in recombination, which is those swapped alleles on the homologous chromosomes. It promotes genetic variation. So another way that you will sometimes see this shown as is with these diagrams of the chromosomes. So you've got lines that represent the chromosomes. So for example, if you have the capital letter D here and the lowercase e right here, that is represented with this chromosome right here, capital letter D and lowercase e right here. This one right here is the sister chromatid to this one. So technically this is one chromosome right here with two sister chromatids, and they are exact copies of each other, which makes sense because they're sister chromatids. And so that bottom one would represent this chromosome right here. This represents the gamete that would be created after meiosis occurs. So we've got a gamete produced that is capital letter D, lowercase e. This is if recombination and crossing over did not occur. Okay, these are the two gametes that would be created from these homologous chromosomes if crossing over and recombination does not occur. Okay. So this is usually what happens if the genes are linked to each other. We say that they're linked to each other if crossing over rarely does happen. And the reasons for this will be discussed more in detail in further subtopics. But essentially, the alleles of the genes, this is one gene, the blue, and then this is another gene, the red, that they each determine different traits, but they are said to be linked to each other if they're very short distance from each other on the chromosomes. If they're a very short distance, they're much, much less likely to go through crossing over and produce gametes like these ones that you see on the right side. Okay, so this is an example of what happens if crossing over does happen between the two different traits and the alleles that are on the homologous chromosomes. So for example, here you have the uppercase D, lowercase e, that's represented right here with this chromosome. Then you have the sister chromatid right here, that would be uppercase D, uppercase e. And this is a recombinant chromosome right here because, as you can see, the uppercase E did get transferred from this homologous chromosome right here, right, to this homologous chromosome. You can see that the sister chromatid on this homologous chromosome got transferred over to this one, and vice versa. They got switched, they swapped places, right? And by the way, these chromosomes that are being shown here, this is one chromosome, this is another chromosome, right? And if you were to see the rest of the chromosome, they're cutting them off, you would see the centromere and the top part of the chromosomes as well. Same here, this is one chromosome, this is another chromosome, but they've already gone through crossing over and swapped genetic information with each other. Okay, and so then the gametes that end up being produced due to crossing over are quite different than if crossing over did not occur, as shown here on the left side. So you can see there are four different combinations of gametes produced on the right side here with the recombinant homologous chromosomes versus these ones which are not recombinant homologous chromosomes. This is somewhat of a review of 3.3, but remember, since you're in HL, you are being held to a higher standard, and you're trying to know all of these details very well, so let's review it again. You've got the lining up of the homologous chromosomes, which we call the bivalence, right? And this lining up occurs during the process of synapsis. That is the lining up. Synapsis is the lining up, okay? Have they already gone through crossing over? Yes, this pair of homologous chromosomes did go through crossing over. It appears that these ones did not. So they're lined up at the equator of the cell, and the homologous chromosomes get separated in metaphase 1. That's very important that you realize that. The homologous chromosomes are separated in metaphase 1. Okay, so you can see. Now, this slide is dealing with the independent assortment of genes that is due to the random orientation of pairs of homologous chromosomes in meiosis 1. So, independent assortment, we haven't really talked about that yet. That is just this idea that the alleles and genes on the chromosomes could end up in any of the gametes, really. They independently assort. They're not connected to one another. They independently assort, unless it's a very rare case where they are linked genes. And they independently assort due to the random orientation of pairs of homologous chromosomes in meiosis 1. So what does that mean? Random orientation means you could have this pink one on the left and the blue one on the right, but you could also have it the opposite, and this does influence the gametes that are produced in the end. Okay, so here we have the homologous pair of chromosomes actually being separated during anaphase 1. They call this reductive division because what's happening? They're going from diploid, being together, diploid, a full set, right? You have all of your chromosomes, all of your pairs of homologous chromosomes, and then all of a sudden you're separating the chromosomes and reducing the number of chromosomes by dividing them apart. And when you reduce them, you're of course creating haploid number of chromosomes. Here we have metaphase 2. It's when the pairs of sister chromatids are now aligned at the equator. We no longer have the homologous chromosomes paired up, right? We just have single chromosomes paired up in the equator with their sister chromatids, right? And so this is when in anaphase 2, once we get to anaphase 2, they're actually being separated and pulled apart. The sister chromatids are being pulled apart to opposite poles of the cell to create that second division, right? That happens in telophase 2, where we now have these new nuclei that are generated, and they're completely different from one another. Notice, if you look very carefully, these gametes do have different arrangements of alleles, different combinations of alleles, promoting genetic variation. So let's check our language, because this subtopic does have a lot of vocabulary and you're going to have to practice it in order to be able to understand fully. So this image shows what? Pause your video. It shows a bivalent, right? A bivalent. Why is it a bivalent? Because it has the homologous chromosomes lined up with each other. Okay, what is this image showing? You can pause your video. It shows one pair of sister chromatids. Okay, so these are sister chromatids, and have they already gone through crossing over and recombination? Yes, they have. So they're actually not exact copies of each other at this point, because as you can see, some genetic information already was exchanged with the homologous chromosomes.